Assalamu alaikum wa rahmatullahi wa barakatuh. Hello everyone, welcome to lecture 1, Organic Chemistry 3. Amino acids, peptides and proteins. Alpha-amino acids are the monomers that make up proteins. Each amino acid has the same fundamental structure, which consists of central carbon, we call it alpha carbon, bonded to NH2 and COH group. Every amino acid also has another atom, we call it R group here, bonded to the central alpha carbon. Biopolymer. The monomeric amino acids are linked through an amide bond, we call it peptide bond. Carboxylic acid of one amino acid with the alpha amino of another one, elimination of water to form the amide bond. If the number of the amino acids less than 50, then the product will be peptide. If number of amino acids linked together via amide or peptide bond more than 50 amino acids, then the product will be protein structure. Classification of amino acids. We can classify the amino acid according to the position of the amino group from the carboxyl group. If it's on the alpha carbon, we call it alpha amino acids. If it's on the beta carbon, alpha carbon, beta carbon, we call it beta amino acid. Here is the carboxylic acid, here is alpha, here is beta, here is gamma. That's why we call this gamma amino acids. There are 20 genetically encoded alpha amino acids of this nature present in protein. 9 of these considered essential amino acids in human. Why they are essential amino acids? Because human body cannot produce them. They are obtained from the diet, from the food we eat. 19 of the alpha amino acids are primary amines, only proline secondary amino acid. 19 are chiral, only one glycine is a chiral because it lacks chiral center or stereogenic center. The natural configuration of the alpha carbon is L. What's L and D? Okay. Recall from organic 1 when we start drawing, taking stereochemistry and drawing the Fischer projection. This is a Fischer projection of D-glyceraldehyde. We have this CHO aldehyde and the CH2OH in the back when we make the model, and the H and OH are in front. So these two will have this line, and the OH will have the right hand side drawing here, OH. So this is a D. The enantiomer mirror image will be L. OH will be in this side, left hand side. Which carbon controls the DL configuration? In sugar, we have the one adjacent to the terminal CH2OH. If that's on the right hand, that's D. On the left hand will be the L. Here is the alpha amino acids. COOH and R will be in the back, and H and NH2 will be on front. Well, so this one will be on the left hand side, and that's why it's L-alanine. It's L-alanine. All common amino acids are L-enantiomer. Their alpha carbon chiral center is the L-enantiomer. Based on the structure comparison with the L-glyceraldehyde. However, not all L-amino acids are levorotatory. Some are actually dextrorotatory. So there is a difference between L and D configuration and the actual optical activity of the molecule. Let's see, we have L dextrorotatory alanine, but we have L levorotatory serine as an example for that. Okay, so there is a difference between the LD and plus minus, dextrorotatory or levorotatory. The RS system of naming chiral center. Go back to organic chemistry 1, stereochemistry part. The relative ranking of priority, SH group, superior than OH, then NH2, COH, aldehyde, alcohol, CHC, and the least one will be the hydrogen. A chiral center has 4 different functional groups. First, you have to identify this function group and arrange them according from higher priority to the lowest priority. View the chiral center down the bond from the chiral center to the lowest priority atom. So we hold the lowest priority in our hand and keep the rest of the molecule in front of us, and then we arrange them according to priority. And then we go, if it is going from highest to lowest clockwise, then it is R. If the priority goes counterclockwise from higher to lowest, then it is chirality is S. Chemists found that regarding S or R nomenclature, nearly all amino acids in protein are S at the alpha carbon. Cysteine is R, and glycine is not chiral. So most of them are S, except cysteine is R, and glycine is not chiral. Alpha-amino acids are classified by the properties of the side chain R group. First group, nonpolar. Here we have H, here we have CH3, here we have isopropyl, isobutyl, secondary butyl. We have here thioether side chain. We have here cyclic CH2, CH2, CH2. We have here benzyl. And we have here this in tryptophan. These amino acids classified as non-polar. We have also polar but non-ionizable, like the one in serine when we have CH2OH. Again, alcoholic functionality, phenolic functionality. Here we have thiol, we have R with amide, another R with amide. They are polar but non-ionizable. We have acidic amino acid where we have two function carboxylic group, like in aspartic acid. So this is ionizable, acidic functionality, COH. Basic functionality, when we have NH2 group, so we have here basic site, we have here basic site, and we have here basic sites in leucine, histidine, arginine. Which categories of amino acid would you expect to find on the surface of the soluble protein? And which would you expect to find in the interior? What distribution of amino acids would you expect to find in protein embedded in a lipid bilayer? Well, polar and charged amino acid residue are most likely to be found on the surface of the soluble protein, where they can interact with water. And non-polar amino acids are more likely to be found in the interior, where they are sequestered from water. In membrane protein, non-polar and hydrophobic amino acid chains associate with the hydrophobic tails of phospholipids, while polar and charged amino acid side chains interact with the polar head group or with aqueous solution. However, there are exceptions. Sometimes positively and negatively charged amino acid side chains interact with one another in the interior part of the protein. And polar or charged amino acid side chain that interact with a ligand can be found in the ligand binding pocket. A chemist is working for helping formulate a new protein powder which includes essential amino acids and is looking for the best amino acid that will readily dissolve in water to produce a smooth protein shake. What part of an amino acid structure should he or she be looking at to determine its chemical nature? Amino group, R group, carboxylic acid group cannot possibly find. Well, the group that control that is the R functional group. So this is a selected correct answer. See you next lecture.